All right, this is question 2, parts A, B, and C. I'll be reading the questions just off my computer here, but I'll be using just this for the notes. I'm taking this on my phone, so let's dive into it. So for part A, our question is, draw the structure of a nucleotide which will include a phosphate, 5-carbon sugar, and nitrogen base group. In your drawing, label the phosphate, sugar, and the base. So here I label the phosphate, and the O here, the sugar, the 5-carbon, and the base. There's some options there. And so here I just labeled the phosphate, the sugar, and the base, and just drew this out. And that's pretty much covers part A. Moving to part B, we have contrast DNA and RNA by explaining how their molecular structures are different. Then explain how their functions in the body are different. So here I have DNA, so DNA versus RNA. DNA has a deoxyribose sugar, while RNA has a ribose sugar. DNA uses thymine, T, as a base, while RNA uses uracil, U. DNA is typically double-stranded, while RNA is usually single-stranded. And so DNA stores genetic information, while RNA is involved in protein synthesis. So there are about 4 reasons contrasting those, and just going over the rest of that question. Moving to part C, our question is, viruses are small particles containing genetic material that can infect any cell type. Describe the structure of viruses and explain how viruses infect cells and replicate. In your explanation, be sure to use each of the following terms: host cell, nucleic acid, caspid, envelope, protein, spikes, and budding. So here we have viruses have genetic material, DNA or RNA, a protein, caspid, an envelope in some viruses. Viruses have genetic material, DNA or RNA, and then a protein caspid or an envelope in some viruses, and protein spikes. And so I wrote down here in 4 different parts here: attachment, entry, replication, assembly, and release. So attachment is when the virus is attached to host cells via protein spikes. Entry is when the viruses enter the host cells, often through endocytosis. And then replication, viral nucleic acid directs host cell machinery to replicate the virus. Assembly, new viruses are assembled within the host cell. And release, viruses are released through cell lysis or budding. And so that just goes through different parts. That just goes through all of the different parts of how viruses spread and infect a host cell. And that is all parts A, B, and C of question 2. Thank you.